Wouter Meuleman, Altius Institute, Functional Organization of the Accessible Genome. So my interest is in understanding the organization of the regulatory genome. And in order to do so, we need to map it first. And one way in which we do that in the lab is to use DNA-seq. DNA-seq basically allows one to identify regions of chromatin accessibility through cleavage of accessible DNA. And so we identify accessible fragments in the genome using the DNase I enzyme, sequence these, map them to the genome, and based on this, we can identify these regions of DNase I hypersensitivity, and we call these DHSs. We've now done this across more than 400 different cell types and states in the human body. I'm showing you here just an example of a small subset of data sets in the hematopoietic system to show you that there's a huge variety in occurrence patterns of these DHSs. Some occur across all or nearly all cell types, whereas others are much more restricted to certain subsets. And so in order to try and analyze these data across all these different data sets, we need to come up with a common coordinate system for DHSs. And so we have developed a simple approach that basically starts off with more than 75 million DHS locations, so DHS peaks, and their summits across all data sets. We project these down onto the genomic axis to find regions of isolated chromatin accessibility, and then we delineated their start and end positions, as well as come up with an estimate of their centroid, or center of mass, and an indication of the positional variability of these elements. And so this works quite well for the very clean cases, but it also works quite well for the less clean cases, such as these DHSs that show a much larger dispersion, or even are a little skewed relative to each other across data sets. So we still get this nice start and end position. We just get a larger positional variability reflected in the wider core region. So now we have this, we can build a matrix such as this one that documents across all the 700 and something data sets which DHSs show up, right? So we expect to see a lot of data around the diagonal where we see similar cell types essentially sharing similar DHSs, but we also see a lot of activity sort of off diagonal. In particular here in this region, we see that there's thousands of DHSs that are showing similar behavior across quite different cellular contexts. And so we wanted to annotate these 3.5 million DHSs and figured that the right way to do that would not be to use conventional clustering approaches, but instead we would need to come up with some sort of a decomposition of this matrix into a much smaller number of components. And that's what we did. Here, each DHS is described by a mixture of 16 non-negative matrix factorization components. Each of these is here represented by a single color. And so these, for instance, these 10 example DHS representations show that there's a huge variety of possible mixtures of components. Some DHSs are primarily annotated with a single component, others show some mixture, and others are annotated with a full rainbow spectrum of components. And we find that this actually corresponds with the level of shared behavior of a DHS across cellular contexts. So for each component here, I'm showing you the DNA-seq data set that's most strongly associated with that component. So we find the yellow component associates strongly with trophoblasts of placental tissues. CD8 T cells are associated with the orange component, and so on and so forth. And we see indeed the DHSs that are primarily annotated with a single component show up in a very limited number of cellular contexts, in this case only placenta, whereas if there's mixture in component space, there's typically mixture in cellular context space. And this is the extreme example here where we see DHSs annotated with all the components showing up across all these different cell contexts. And so we use this information of which data sets are most strongly associated with each component, along with which transcription factor motifs are most strongly enriched in DHSs in those components, to come up with an initial labeling or a DHS vocabulary. Now, to simplify things a little further, going forward, we're simply just going to assign every DHS to a single component, namely its majority component. And so now we have on the one hand a DHS index with more than 3.5 million DHSs with a lot of annotation, positional annotation, but we also have annotation in terms of their behavior across different cellular contexts in terms of this DHS vocabulary. We can now use this to essentially re-annotate the regulatory genome. And so, for instance, if we look around GATA1 gene here, we zoom into that, we see that there are a lot of DHSs that are all annotated in the same component, namely the myeloid erythroid component. And that's kind of striking because the GATA1 gene is also plays a very prominent role in the erythroid lineage. And so we figured whether this is something we could exploit to maybe start labeling genes based on their regulatory landscape. So we systematically tested all of GenCode annotated genes for association with these DHS components, and we find that this very often works quite well in terms of, for instance, FOXP3, an immune-related gene being annotated in the lymphoid component, and so on and so forth. And so this works well across the board. I'm showing you here just a couple of examples in terms of the top 5 genes annotated for these components. In particular, for transcription factor genes, we find that for the cardiac component, for instance, the top 5 genes are essentially all the lineage-specifying factors that we recover. So completely agnostic to any kind of gene expression data. We can also use these data to annotate genetic variation and trait association. And we find that DHSs annotated with specific components much better capture the heritability signal than unannotated DHSs or other genome-wide annotations. We find that this is the case for more than 260 traits in the UK Biobank. In particular, if you focus on this core region of these DHSs, further emphasizing that the delineation aspect of this work is very important. And so we have a common coordinate system for regulatory DNA with a component-based annotation. We use this for characterization of gene regulatory landscapes and genetic trait association. And interestingly, we can now also use this to sort of project in new data sets for which it's very hard to do de novo p-calling, such as single-cell assay data. These are the people that help with the work. The paper browser and data are available here, and I urge you to also use our newly developed browser at this URL. Thank you.